Hi, my name is Sarah Moehring. I'm a senior biology major here at California University of Pennsylvania.
Hi, my name is Maenzie Cree, and I am also a senior biology major here at CalU.
Today we are going to present to you the utilization of organism classification in the advancement of abandoned mine drainage treatment. For some background before we begin, abandoned mine drainage comes from water that has been polluted by waste created during mining activities. Because the state of Pennsylvania produces 25% of all the coal mined in the US, this is an issue that is very relevant for us. Scientists have estimated that over 4,000 miles of rivers and streams in Pennsylvania are considered biologically dead from the byproducts of coal mining. Boyce Park has a passive remediation site located in Plum, PA. This is where we extracted our acid mine drainage samples in order to be tested. It has a low pH of around 3, and its primary contaminants include things like aluminum, iron, and sulfate.
For our hypothesis, we hypothesized that as our samples were collected from Boyce remediation site, that the purified isolate would play a role in acid mine drainage remediation.
In order to test this hypothesis, we performed serial dilutions of the acid mine drainage samples that we collected from Boyce Park. Then we created streak plates in order to make overnight cultures. Next, with those successful overnight cultures, we performed a qubit assay and then ran PCR. During the PCR, we extracted DNA. When we had that extracted DNA, we then sent it out to companies in order to allow it to be sequenced so we could gain more information.
For the results of our experiment, we found that our DNA concentration was 1.4 micrograms per milliliter. In this experiment, we used de novo sequencing in which there was no reference sequence. So we started at the bottom of this first diagram, and we started with about 6.4 million reads. And then we were able to identify the contigs, which are the repeated sequences, and we were able to assemble these contigs into a single genome that could be studied. It was found that this isolate was most closely related to Elizabeth Kingia anopheles, and it was related about 85%. So then we decided to run the sequence, the 16S in BLAST, and it matched 99% with Elizabeth Kingia meningoceptica. And it was found that in scientific research articles that these two isolates are commonly mistakenly identified. So that is why we saw that in our results. We also found that there was no scientific research that supported that it played any type of significant role in acid mine drainage. The research did show that it has been found in hospitals, in their water supply and their medical devices, such as catheters, feeding tubes, and respirators. And this bacteria is most commonly known to cause meningitis in newborns. Lastly, we found that it possessed antibiotic-resistant genes. It has the BLA genes, which encode for beta-lactamase, which is an enzyme that breaks down the antibiotic ampicillin. And as the diagram shows up at the top, you can see where the BLA gene is the green arrow. And it can be inferred that horizontal gene transfer can be occurring, which would induce these antibiotic-resistant genes into new organisms that could also be found at Boyce Park.
To wrap up our research, the isolate was found to have antibiotic resistance genes, which allowed for us to form a second hypothesis that could shape a future direction in which we would further this research, that the horizontal gene transfer method could play a role in the gene characteristics found in Boyce Park. This allows for the conclusion that there is no correlation between the Elizabeth Kingia meningoceptica and acid mine drainage in Boyce Park in the end. Thank you for coming to our talk.